check out the glowing nematode, glowing green, and the red and green zebrafish. How do they glow, do you ask? Well, they express a protein. One of the proteins is DS-red, and the other is GFP. GFP was isolated from jellyfish, and DS-red is from coral. And what's interesting about these two proteins is they're from two different organisms, but if you match up their amino acid sequences, it's only 24% identical. But what's really interesting is, let's check out their structures. So one way you can get them is by going to the NCBI website. Should be the first link when you Google search it, and then select structures, and then search for GFP. Then I'll just pick the first one, the first structure, and then click on the PDB link. And here we're going to want to get the PDB file, which is just a text file that has all of the X, Y, and Z coordinates for all of the atoms in the structure. So we'll just go and save that. Now let's go and get the structure for the DS-red, the red protein from coral. But a lot of these are just with one of the molecules or with two. So let's get the one that is the tetramer version of the structure. So DS-red exists. It's the same molecule, but there's 4 of them that are associated together. So again, let's look at the PDB file. So I want to show you something real quick. You just scroll down. So when you get to the part that shows all the coordinates for all of the atoms, where is it? Right, so you see those that have an A, that's for the A chain, one of the 4 chains. And then the B chain. And then the C chain. And then the D chain. So we'll just save that the same way as I did before. So now you can just look at those files under the a program called SPDBV, and you can get it from that website there. So let's just open up one of the files that we saved, and it saves it as a text file. So you'll have to just search for all files, and it should come up. So here's our structure, just the raw structure. Kind of hard to make anything out of it. So we'll just go to the control panel and change it up. Get rid of all the side chains. So here we can just see the carbon chain, backbone of the protein, and we can highlight the ribbon so it give it a green color. So this gives the backbone a kind of a ribbon color, so it kind of makes it easier to visualize the structure. We'll just go ahead and get rid of that backbone, and you can also kind of give it a 3D look. So that's the structure for GFP. So let's open up a program again for the DS-red. this is the file that we saved. So again, it's really hard to visualize what we're looking at here. Kind of see the four chains. So let's go ahead and get rid of the side chains. Still kind of hard to visualize what we're looking at here. So let's visualize with a ribbon. Switch it over to give it a red color. Now I can kind of start to see it a little bit better. I'll get rid of the backbone. So now it's a little bit easier to see the four different molecules. And what's interesting about this, I'll just show you this in a minute, that all of those four molecules have the same structure. It's just they're put together to form the tetramer. They're just kind of orientated together differently, but they're the same structure. So here's a video that I found on YouTube. It kind of shows the structure for DS Red pretty good. So let's go ahead and open them up together. So again, let's go back and open up the file for DS or GFP. Do it the same way as I showed you before. So get rid of show the ribbon, get rid of the side chain and the backbone, give it a green color again so we can tell them apart. Now it's open, and I've made a modified file which I've just gone and deleted all of the pieces for the A, B, C, and D chain. So now it's just showing one of those molecules for the DS Red. So I'll go and get rid of the side chains. Give it a red color. Get rid of the backbone. So you can see the monomer for the DS Red and the GFP structures together. So the next thing we're going to do is do magic fit. So what this is going to do is it's going to superimpose the two structures on top of each other. So remember, these two molecules, these two proteins, are from two different species. One's from coral and one's from jellyfish. And what's really interesting about this is that even though they're from two different organisms and they both produce fluorescent proteins of different colors, their basic structure is pretty much spot on. And this is remarkable because remember, the amino acid sequence is only 24% identical, yet their structure is very close. Also, another thing too is they've produced variants by just mutating these genes, so they have a whole spectrum of different colors.